Thank you, Mr. Chairman, and thanks to the organizers for inviting me. And I would like first to emphasize the first three words of the title of my lecture, which is ultra-low doses of cannabis. And when I say ultra-low, I mean really, really low doses of cannabis, much below the dose which is required to get the acute, well-known effects of cannabis. Will it be an euphoria, a psychotropic effect of analgesia? I am going to talk about doses lower by 1,000 times less. And then I'll try to convince you that these low doses of cannabinoids can protect the brain, can protect the heart, and can protect the liver from all kinds of insults. And I'll show you some experiments with animals, and I am completely aware to the fact that most of you are not biologists, so I'll take it easy. I'll go slowly and give you only the essential data, not all the details of the experiments, and then we shall discuss some medical and other implications of the finding, and I'll try to convince you that by using the ultra-low doses of THC or other cannabinoids, but THC, that's the main phytocannabinoids, we can treat a variety of pathological conditions from liver transplantation to heart attack, from stroke to trauma to the brain. And then we'll discuss what is the best cannabinoid preparation to use for all these treatments. So let's begin with the brain, simply because we know a lot in the literature. There is a lot of information about neuroprotective effects of cannabinoids. Here we have some of the leading works in the literature showing that all kinds of cannabinoids. Here we have WIN and CP. These are synthetic cannabinoids. The THC, the main psychoactive ingredient of cannabis. This is the anandamide that we heard a lot before, and 2-AG, the two endocannabinoids. All of them protect the brain for all kinds of insults, beginning from ischemia, going to WABA intoxication, or traumatic brain injury. In all of these cases, various cannabinoids protect the brain. And there are two common features for all these studies. One of them is the dose that was used in these studies. All of them used the conventional dose, namely the dose that it's known to affect in rats and mice. That's the animals that were used in all these studies to activate the known effects of cannabis, analgesia, appetite, and so on, which is between 1 to 10 milligrams per kilogram. The other common feature is the therapeutic time window. In all these experiments, the cannabinoid was injected few minutes before or 2-3 hours after the insult. If it was delayed, the treatment was delayed, it was not effective anymore. I'm going to talk about a completely different phenomenon. In our case, first of all, as I told you, we are using much lower dose of cannabinoid, the THC, 0.002 milligram per kilogram, compared to the 10 milligrams that other studies used. The second difference is that we are going to inject the THC 1 to 7 days or 1 day after the insult, not just few minutes before that. And then we'll measure the effect of the insult few weeks later. And it means we are dealing with long-lasting effects of the cannabinoid, of the small amount of the cannabinoid. We use all kinds of insult just in order to make sure that we are not dealing with a specific phenomena, but with a more general phenomenon. We'll use deep anesthesia, neurotoxicity of ecstasy, hypoxia, which is produced by exposure to carbon monoxide, and epileptic seizures which is used by PTZ, a material which is known to cause epileptic seizures. All these conditions are known to cause behavioral deficits or cognitive deficits in human beings as well as in rats and mice. And then the way to measure the cognitive deficits in animals is by using behavioral tests, and we use a battery of tests again to make sure that we are dealing with a more general cognitive deficits, not a specific one. And I'll just bring you two small examples for that. Two behavioral tests to measure the cognition of the animal, the functionality of the cognitive processes of the animal. Let's begin with just one example. This is a well-known behavioral test which measures spatial learning of mice. The mice have to find a location, a certain location in the field where it gets the water. In the beginning, the mice has to travel along the whole field till it hits the well, the hole with the drop of water, and drink the water. And with time, the clever mice learn how to find the best way to get there, and they get to the water in a very short time. So this is one way to dissociate between the so-called clever and stupid, or damaged or deficited mice. So here we have one of the experiments. Just don't get confused with all these dots. Follow, please, the black line here. In the beginning, it takes to the mice 150 seconds to find the water in the field. And with time, with repetition, with more and more trial, it learns and get to the water, let's say, in 60 minutes. Now there is another curve here in purple. These are mice that few weeks ago were treated with metrazole, the drug that causes epileptic seizures. And this drug and these mice fail to find the water, or fail to learn the best way to get to the water. It's easier to look at this presentation. These are the control mice, and these are the mice that were damaged by epileptic seizures few weeks ago. Now, however, if we inject the very low dose of THC 3 days before the seizures, we get improvement. We prevent the damage. So this is the damage, this difference, and if the mice receive THC before the PTZ, before the epilepsy, it behaves almost like the control mice. We can repeat the experiment again and again. We can inject the THC either 1 day or 3 days or 7 days before the PTZ. And in all these cases, we see the damage to the purple, the damage of the PTZ, of the epilepsy, here and here, and the protection by THC, pre-treatment or by THC. I would like to bring another example of another test. This is the object recognition test, which has nothing to do with spatial memory. This test is based on the natural behavior of the mice. Mice are curious. They always prefer novelty. They prefer to spend time with new stimuli rather than well-known old stimuli. So we put the mouse in the arena with two identical objects, and the mice are sensing, smelling, touching the objects. And the next day, we put the mouse within the same arena, but this time we replace one of the objects and we put a new object. The normal mouse is curious, is interested in the new object, and spend more time here. So they're assuming that he still remember what was the object yesterday. So this is a very elegant and nice way to distinguish the one mice that remember and fail to remember what was in the field yesterday. So here is an experiment, for an example. These are the normal mice. This is in black, you have the new object. The normal mice prefer the new object rather than the old object. mice that were treated with PTZ, namely with epilepsy, few weeks ago, they failed to dissociate between the two objects, as if they don't remember which is the new one and which is the old one. However, if they receive THC 2 days before the PTZ, they behave almost like the control animals. So these were just 2 examples, and I can give you many more, but I'll not do it. I save your time. So here we have just, I put together some graphs, all kind of insult. This is the pentobarbital deep anesthesia, which is known to cause deficit damage in human beings and in mice. This is the ecstasy experiment. This is the PTZ, and this is the hypoxia. In all of them, the THC protected the brain from the damage. However, I would like to draw your attention to another finding here. In some of the experiments, you may notice that THC by itself causes what appears to be a slight damage, a slight deficit in memory. This is not the case here, but this is the case here. This is not the case here, but the case here. So it means sometimes there is a slight, although this difference is not significant. And this led us to another idea, and with your permission, I'll leave aside the story of cannabis for 30 seconds and tell you another story, which is very important in biology. And then we'll come back to the cannabinoid story. And the name of the story is preconditioning, or hormesis. Preconditioning was discovered about 3 decades ago by cardiologists, and they found that if one cause a very delicate ischemia to the heart is more resistant to massive ischemia that follows. So they call the phenomenon ischemic preconditioning. The slight ischemia protect the heart. Then the neurologist found the same phenomenon in the brain, namely a very delicate ischemia to the brain protect the brain from a following massive ischemia. The next part of the story is that this is not true only for ischemia, but for other insults as well. For example, hyperthermia, elevated temperature. Elevated temperature causes a lot of damage in the brain. However, if you first elevate the temperature of the brain very slightly, without causing a real damage, this brain becomes resistant and can tolerate massive hyperthermia later on. This is the phenomenon of preconditioning of hormesis, and then it was found that there is cross-conditioning, so one can use the delicate ischemia to protect against hyperthermia, or delicate hyperthermia to protect against the ischemia. And of course, biologists became very interested in this phenomenon, and they look for the molecular mechanism within the brain and within the heart, and they found several molecules that take part in this protecting mechanism, the endogenous protective mechanisms of preconditions. So there are a few names here for those of you who are interested in the name. I'll just mention 2 of them. CREB is a transcription factor, namely a molecule that regulates the expression of gene in the cell. And this is BDNF, which stands for brain-derived neurotrophic factors. This is one of the nerve growth factor, a molecule that promote the survival and the development of neuronal cells. So these two molecules were found to be involved in the general phenomenon of preconditioning. So now we are going back to the cannabis story. We were interested to know whether any of these molecules take part in the protection that is induced by the very low dose of THC. And here are the results. The experiment was done like that. We inject the low dose of THC. We wait 7 weeks, and after 7 weeks, we looked into the brain, and we looked into 2 specific regions of the brain, Those of you who are not neurologists, I'll just tell them that hippocampus and frontal cortex are two major parts of the brain that are essential and crucial for all the cognitive activity of the brain, memory, learning, and so on. And in these two important areas, we find elevation of ERK, elevation of CREB, and elevation of BDNF, the three molecules that are known to protect the brain 7 weeks after a single treatment with the ultra-low dose of THC. So now we can summarize these results in a very simple cartoon. The various insults emerge to cause neuronal damage, and the neuronal damage causes long-lasting cognitive deficits. Then we can see from the outside, measure by the behavioral test. But of course, and now when we inject THC, we activate the molecular signaling mechanisms of, in this case, CREB and BDNF, which protect and prevent the neuronal damage. This simple cartoon is oversimplified because a lot more is happening in the brain during the damage. And the most important process that happens is the reaction of the brain to the damage. And this reaction is carried by the immune system. As a response to the damage to the neuronal tissue, the immune system is mobilized, and cells of the immune systems, either resident microglia within the brain or macrophages that arrive from the circulation, arrive to the area of damage, and they cause inflammation, local inflammation, by producing and secreting all kinds of molecules. One of them, the most well-known, are the prostaglandins. These molecules are supposed to protect the neuronal cells, but unfortunately, they by themselves cause damage, secondary damage. So this is the secondary damage which is caused by the inflammatory response of the brain. So we have here a positive feedback circle. So the first damage mobilizes the immune system that by itself causes a secondary damage. So we wanted to see whether the ultra-low doses of THC can also protect the brain from the secondary damage. So for that purpose, we eliminate the primary damage, and we activated the immune systems directly, and there is a known trick to do it. For the non-biologists, I'll explain that this is a bacterial lipopolysaccharide. It's something that is triggering the immune system. Once we inject it, the animals or the human beings become very sick. The immune system is mobilized, and we have total inflammation, and the animal appears very sick. It has fever, but after 2-3 days, they recover, as it happened with us. They recover, and they appear happy and healthy. So we wanted to see what happens if we activate the immune system without the primary damage, and this is the experiment that we are going to perform. We inject the LPS for the first 2 days. The animals are pretty sick, and then they recover, and after 5 weeks we measure the cognition or the cognitive functioning of the animal. And of course, we expect them to be okay because they recovered already. Unfortunately, we find if this is once again, this is the object recognition test. I'll remind you that if they dissociate between the old and new project, it means they remember, and if they fail to dissociate, it means they don't remember well. So in spite of the fact that the animals recover from the systemic disease, there are still deficits in the brain, some deficit in the brain. However, if we inject THC 2 days before the LPS, they behave like the normal animals. We can also inject the THC after the LPS and still protect the animals. That's what is called post-conditioning. What is even more interesting, we can inject the THC even 7 days after the LPS, long after the mice recovered from the systemic sickness, the systemic disease. Still, the animals are protected, as if something is going within the brain along this week. After the animal recovered, a process of deterioration is still taking place in the brain, and we can stop it by THC. Do we have any evidence, any biological evidence for such a process? So, we biologists like to look at molecules, and I remind you that one of the molecules that are activated by the immune system are the prostaglandins, and I'll remind to those of you that know, or I'll tell the others, that prostaglandins are formed from arachidonic acid, and the enzyme that produce prostaglandins is called cyclooxygenase. There is COX-1 and COX-2. COX-1 is not related to our story. COX-2, this is a well-known enzyme because many medicines are working to control the activity of COX-2, including aspirin. That's the way we recover from fever, by giving aspirin that block this enzyme and block the production of prostaglandins. So now we can use the prostaglandin or the COX-2 as a measure, as a marker for the inflammation in the brain. So we go back to the brain now, and we inject THC, the small amount of THC. We wait 2 days, then we inject the LPS in order to activate the immune system, and 7 weeks later we go and check these 2 brain regions that are crucial for cognitive functioning, the frontal cortex and the hippocampus. And what we found, that THC by itself is doing nothing to the brain. LPS, the animal that were sick for only 2 or 3 days, 7 weeks ago, still there is a lot of COX activity in the brain. However, those animals that received THC 2 days before the sickness, before they got LPS, they are protected. The level of COX is almost normal or completely normal in these regions, as if THC prevented the inflammation in the brain and prevented the damage to the brain. Those of you who are interested in cannabinoid pharmacology, we heard a lot this morning about CB1 receptors and CB2 receptors. THC, tetrahydrocannabinol, is not a selective drug, so it acts either through CB1 or through CB2 or through many other mechanisms, what I call here non-CB1, non-CB2 mechanisms. And we wanted to see what receptor mediates the protective effect of THC in the brain, and we have an arsenal of drugs to tackle this question. First of all, we can use selective agonists. Here we have one in green. One of them is working on CB1 but not on CB2. The other one is working on CB2 and not on CB1. Another way to answer the question is to use antagonists. This one blocks completely CB1, so leave the CB2 intact. And the other one blocks CB2 and leaves CB1 intact. So by playing around with all these drugs, we can find which receptor is involved, and this is, of course, very important if we intend in the future to develop more specific drugs rather than using the non-selective THC. So here is one experiment. This is the normal animals, they remember. LPS, animals that were treated with LPS, they failed to remember. This is the protection by THC. However, if we block CB1 by the antagonist, we lose this protective effect, which is not the case if we block CB2. It means CB1 is involved in the protection. And the complementary experiment is done by selective agonists against one. This is the damage by LPS, the protection by THC, and this protective effect is mimicked by CB1 agonist and not mimicked by the CB2 agonist. So we can conclude by saying that CB1 receptor within the brain is the receptor that is very important for the protective effect of cannabis. So let's summarize the first part of my talk. You saw that a single treatment with a very low dose of THC can protect the brain for a long period of time, up to 7 weeks. We saw that in parallel to that, the very low dose of THC activate signaling processes that are known to protect the brain-derived neurotrophic factor and the CREB, and also it inhibit or suppress the inflammatory response as is measured by the COX. And we also saw that the CB1 receptor is the one that is important for this protection. Now, in my title, I promised you to move then to the heart. So let's do it very quickly. Now we'll try to see whether the ultra-low dose of cannabis or THC will protect the heart from damage. So this is the experiment. We take the heart and we cause damage to the heart. The damage that we know is by stopping the circulation to the heart. That's what happened in heart attack. So we ligate the left anterior coronary artery. It means the artery that supplies blood to the cardiac muscle, and we cause infarct, damage to the heart. And then 24 hours later, we measure the outcome of this damage. We can measure by echocardiography. Echocardiography, that's the same as we are doing with patients. We measure the contractility of the heart in vivo, in the animal. In the human beings, we can put it in the machine and measure how well the muscle of the heart is contracted. We can open the chest and look at the brain, at the heart, and measure the size of the infarct. We can measure neutrophil and TNF alpha. These are markers for inflammation in the brain. And we can measure troponin, the serum. This is measured by any laboratory test when the physician is sending you for a checkup. This is what they measure in order to know whether the heart is working well or not. This is a specific, a selective marker for the function of the heart in the serum. So now we are going to compare animals that receive THC, once again, the very low dose of THC, 2 days before the ischemia, to those that did not receive the THC. So I'll begin with the more simple things. This is the size of the infarct, and this is the average in 10 animals that underwent the same treatment. This is the size of the infarct, and this is the size of the infarct, much smaller infarct in animals that receive THC 2 days before we stopped the circulation to this area of the brain. We can measure the contractility of the brain. This is the contractility in black, the contractility of the heart, excuse me, in the normal animals. This is what happened after we ligated the coronary artery. There is a drop in the contractility. However, if the mice receive THC before, it is protected from this drop in activity. And the same with the neutrophils and troponin T. Now, I can show you more and more experiments, but I would like just this one because actually we have here the same results. The only difference that here THC was applied 3 times a week for 3 weeks before the experiments. And the reason for doing this kind of experiments is to show that the protective effect does not undergo desensitization. No desensitization or tolerance or tachyphylaxis. These 2 words means the same. It means that the effectiveness of the drug very often disappear if we repeat using the same drug. This doesn't happen if we use this minute, these very low doses of THC. It means that we can expect to use the low doses of THC for chronic treatment, not only for acute treatment. Okay, then I promise you to talk on the liver, so let's do this. Here we are causing damage to the liver, again, by ligating the arteries that supplies blood to the liver. This is a main medical condition which prevent or spoil some of the transplantation trials in liver transplantation. Now, there are two kinds of damage while you stop the circulation to the liver. One of them, the ischemia by itself. If you stop the circulation to the liver, there is a need for oxygen and metabolites, and there is some damage. What is even more important is the reperfusion damage, because once you open again the artery, it means when you connect back the transplanted liver to the acceptor body, there is a huge flood of oxygen to the tissue, and this by itself causes a massive damage to the liver. So this is the reperfusion damage, which is very important in the case of liver transplantation, and we can measure the damage by measuring all kinds of molecules in the liver. All of them are related to the inflammatory response in the liver, the DNF, or the interleukins, one alpha and one beta. We can also measure selective markers in the serum. Again, when you go for a clinical checkup, that's what you get in the so many results that you have on the sheet of the laboratory. The two enzymes which are selective enzymes that represent what is going in the liver. It means if the liver is damaged, these enzymes, the level of these enzymes is elevated in the serum, the transaminases. So here is the experiment. We inject the very low dose of THC, and then we produce the ischemia and the reperfusion, and we measure 6 hours later what is going in the liver. So let's begin with the transaminases, the enzymes that are present in the blood and represent what is going in the liver. This is the control animals, the vehicle or THC, no difference. Very low levels of these enzymes in the blood. However, if we cause damage to the liver, there is a huge elevation of these enzymes in the serum, the alanine transaminase and the aspartate transaminase. However, if we treated the animals with THC before the damage, before causing the damage, we prevented this elevation in the enzymes, showing that the damage in the tissue of the liver is much smaller than in the other animals. Of course, we can look at the liver itself, and again, don't bother yourself with all the names here. All of them represent inflammation in the tissue. So THC is doing almost nothing, but ligating the artery causes elevation of all these markers, and this elevation is prevented in those animals that receive the ultra low dose of THC. So now I hope I convince you that all these three systems can be protected by THC. And now let's see what is the medical implications of these experiments, because all the experiments were carried out on mice. So the liver transplantation, that's exactly what was mimicked by our experiment with the ligation and opening of the blood supply to the liver. We also saw that we can use the injection of THC either before or immediately after. So we can not only treat animals or human bodies before something happened to them, but we can treat them after, like in hepatic hepatitis or things like that, or heart attack. We don't know when the patient is going to get heart attack, but we still have 1 to 3 days time to treat the patient. I showed you that the effect doesn't undergo desensitization, so we can think about using chronic manner of treatment. In the case of brain, of course, stroke and trauma, it's a well-defined situation where there is some damage to the brain. But sometimes we don't touch the brain, we touch the heart. But if we put the patient for air surgery and put it on a lung heart machine, it means we stop the circulation and connect in order to treat the heart. We connect the circulation to the machine. Very often we cause unfortunately we cause some cognitive deficit to the patient. So before going to this operation, we may treat the patient with very, very low doses of THC, 1,000 times or 100 times less than cause the acute effect of THC, and protect the patients. Now the next question is what preparation should we use? Of course, one can use pure THC, and there are drugs on the market. Marinol is synthetic THC, which is sold on the market. The conventional medicine use it only for rare occasion of sometimes for increasing appetite, promoting appetite in AIDS patients, or as anti-emetic drug in chemotherapy. But this drug is not popular anymore. But of course we can use Marinol. We don't have to use the full dose of Marinol. 1% of a single pill is enough in order to initiate this protective effect in the brain or in the heart or in the liver. Now, what about using medical cannabis? Most of us prefer to use the medical cannabis, the plant, not the synthetic THC. Can we use medical cannabis? And the reason for raising this question is the very fact that medical cannabis, or Sativex, which is an extract of cannabis, contains more than just THC. And the most important, and we heard about that a lot before, is the CBD, the cannabidiol, and there are a lot of interaction between CBD and THC. Sometimes they are antagonizing each other. Cannabinoid can prevent some of the effect of THC. Sometimes they are potentiating each other. So it's called synergism between the two drugs. So would cannabidiol, which is present in medical cannabis, prevent the protective effect of THC? So the last experiment that I'm going to show you, promise you the last one, this is the control animals. They dissociate between the new and the old subject. This is the damage by LPS. This is the protection by the low concentration of THC. Cannabidiol by itself fails to protect the brain. It doesn't protect the brain. But when we put CBD and THC in 1-to-1 ratio, CBD is not preventing the protective effect of THC. Somebody will say maybe there's even potentiation. I'm not convinced by that, but for me it's enough to say the CBD is not preventing the protective effect, so one can use the cannabis, not only the synthetic Marinol. But still, we are left, so we can use the cannabis. Again, we don't need the full dose of the cannabis. 1% of the dose, the regular dose, is enough for that. Still, we are left with the administrative restriction, the legal restriction. In most countries, we are not allowed to use it. There are only few countries. And I hope the Czech Republic will be, as others say, the next one that will allow the use of medical cannabis. But in most countries, it's not allowed. Do we have any other solution? And the solution is here in the outside. Can we use industrial hemp for that? Industrial hemp, according to the EU definition, is the same plant, the same cannabis, but with very low dose of THC, and the limit, the administrative limit, is less than 0.3%. Less than 0.3%, it's 10% or 1% of what we find in the medical cannabis, which is much below the dose that is required in order to get the high or the psychoactive effect. That's why the users are not using the hemp. But it's perfect for our purposes. So if we want to overcome the limitation of the administration and the illegality of medical cannabis, I suggest that we should run some clinical experiment with the so-called industrial hemp. And by that, I would like to thank the people who carried the experiment in my lab in Tel Aviv, and our collaborators in the Department of Cardiology and the Department of Pathology in the Sheba Hospital, and to thank you for your attention.
Zde máte ještě nějaké dotazy? Questions?
English? Okay.
Yeah, I'm sorry. My name is Simon Smith from the Netherlands. I wanted to know, you speak about ultra low doses. This protective mechanism, does it only works with the ultra low doses, or, I mean, there is a lot of people here who smoke marijuana. Does it also offer any protection?
Okay.
Yeah.
We of course, we try to test this point as well. There is some protection by high doses. High doses are less effective than the low doses. And this is the case in biology and medicine in general. It's not true that the more is better. Sometimes higher doses are worse. Actually, I even not sure that the high dose is protecting directly, because we inject the high dose and then we wait 1 or 2 days and we introduce the damage. During that time, the high dose became very low in the brain. So it might be that the low dose that followed the high dose is the protective one. But as a matter of fact, the high dose is less effective than the low dose. But you still can see some protection by high doses. But you don't need it, of course. If you don't want to get the side effect, which sometimes that's the main effect, you want the patient to be high. You want him to still drive safely back home, so you don't have to give him the high dose. Okay, thank you.